Okay, so last day we talked about cells, right? The idea of all the different scientists who helped to accomplish things. So Schleiden and Schwann and Virchow and Robert Brown and Hooke, right? Today we're going to talk about the technology that they had that allowed them to see cells in the first place, microscopes. Okay? If it wasn't for microscopes, we would not be able to see the cell. And I mean, I don't know that this piece of information on the slide right here is very noteworthy, but it says the cell itself is very small. It can't be seen with the unaided eye. Okay. One of the words we're going to talk about over the course of the next half hour is the word resolution versus magnification. Magnification is when you zoom in on something to be able to get a better picture of it, right? All of your cell phones, you guys know how if you just take the two fingers and you do this, right, where you separate your fingers, you can zoom in, right? That's magnification. But the other word we want to use is something called resolution. Resolution is your ability to distinguish two consecutive points. I'm going to say that again because this is a really important concept. Resolution is your ability to distinguish between two consecutive points. Right now, I see two students in front of me, and I can clearly see the differences between you. However, if I were to, say, go on top of the roof of our building and look down at students, I might not be able to tell these two students apart because the further zoomed out you get, sometimes you can't actually distinguish between things. Does that make sense? As another example here, humans have the ability to distinguish things basically as small as hair. Anything smaller than hair, you can't actually see the differences between it. Right now, so I'm just going to use you as an example, I can see individual hairs on your head, but only because I'm like 1.5 feet away from you. Right? I can't see the individual hairs on your head way back there. I know they exist, right? But I can't actually see them. That's resolution, the ability to see things, and this is really the key words right here, in detail. By us being able to see things in detail, we've discovered a whole new branch of biology. And we'll discuss this at the end of the lesson, but because of this has led to the field of genetics. You guys remember genetics, the study of genes? We're going to talk about DNA and how by us being able to see that, as well as things like viruses and cells, it's really created a boom in what biology does. Okay? Here's where I'm going to start. This picture slide right here. This right here is a picture of salt with 3 different magnifications. The top right here is basically just normal, everyday, I can see salt. And salt is actually, unless you get really close to it, you probably can't see individual grains of salt from far away, right? Oh, I didn't mean to draw that on there. Let's erase that. Eraser. Okay. One of the best examples I can give for how resolution works is pixels. You guys know how a pixel works on like an image? Is that like right about here, I'll see that I can kind of zoom in on it. Right about here, I think there's like a pixel, like a square, right? And different screens have different pixelation amounts to them, right? You guys ever heard of like 1080p or 4K resolution? The ability to have all these different pixels? Well, this salt right here, I cannot tell one salt molecule from another. It's not possible. But if I were to zoom in with a light microscope, which is what this one right here does, a light microscope, I can actually see individual grains of salt inside here. Now, in terms of this individual grain of salt, though, I can't really tell you much about what's on the grain of salt or what it's made out of, but I can tell A apart from B. So I do have the resolution to tell them apart. But that's not nearly as good as this. This one down here is the electron microscope. And on the electron microscope, can you guys see how there's even like details on the surface right here where I can actually see like some sort of like surface texture to it? Okay. That's your resolution. The ability not just to zoom in, but the ability to zoom in and see details. That's what you need to have. Okay. Here's your basic light microscope. How does a light microscope work? It's all about optics and lenses. Okay. Inside your basic light microscope, there are 2 lenses. There's one lens right here, and then there's a second lens right here. And what these lenses do is they bend light. Okay? It's really no different than what your glasses do, if any of you wear glasses or contacts. It refracts the light, it bends it, and if you know enough about the principles of optics, it's like a magnifying glass. You can make something bigger. If you have 2 different lenses, what ends up happening is light goes from the light bulb underneath the microscope, it goes through this lens, it goes through this lens, and when you put your eye into the piece up here, you see a zoomed in image. Now, one of the cool things about light microscopes is that they give you the ability to zoom in to about 1250 to 1500 times magnification, which is a lot of zoom. Like that is considerably more than what you can see without a magnifying glass. Here is a picture of just your generic microscope. And I don't need you to memorize all this terminology. Like, I'm not going to test you on what is this called, what is this called. You can look that up. But I want to discuss just some of the basics of what's in a microscope that we'll use on Friday. First of all, there is a lens called the ocular lens. It's basically this tube right here. And somewhere inside this tube, there is a piece of glass, a lens, and it probably multiplies everything 10 times in terms of its zoom. It lets you zoom in 10 times. That's what's traditional. It might not be 10, but it probably is. That's one lens we have. But that's not the only lens we have. These are often known as compound microscopes because you get these little rotating objective lenses right here. And I wish they'd have wrote on this particular slide, this is the objective lenses, because there's 3 of them. And you rotate and click them in depending on which one you want to use. Most of our microscopes come with a ×4 lens, a ×10 lens, and a ×40 lens. But that doesn't have to be the case. That's just what most of our school microscopes do. Here's how that works then. If you have a lens in here that's ×10, and you have a lens over here that's ×4, it's a zoom of 40. You multiply. 10 by 4 is 40. If you use the 10 with the 10, it's 100 times zoom. And if you use the 10 with the 40, it's 400 times zoom. Most of the microscopes in our school are 400 times zoom is the best you can get. There are some lenses, I think there's one in this school. I had one back when I worked at Walsh, where there would be a 4th objective lens on here. And sometimes you can get one that's so big it's at ×100. 10 by 100 means that you could possibly have 1,000 times zoom, which is pretty incredible. Usually those microscopes run in $1,000 range. Now this then leads to the question, Chris, why do we stop here at 1250 by 1500? Right? Like, why can't I just use an extra lens? Why don't we use another tube? Why don't we have an extra one right here and make this one be by 10? Why don't I go 10 by 10 by 100? Why don't I go 10 by 10 by 10 by 100? Why don't I just keep adding lenses and keep zooming in? And that's the problem, is that we're discussing 2 different things. There's the ability to zoom in, sure, but there's also resolution. If you can zoom in, but you can't actually tell the difference between this and this, if it's just all one big blurry blob, just because you zoom in doesn't mean it's helpful. Okay. And one of the best ways of me describing this is through photos of pixels and stuff like that. So before I get to that, though, let's discuss this term first here, magnification. Okay. Magnification is your ability to zoom in, and it's all about this word right here, refraction. Okay. We talk about this for like 1 minute in Science 10, but if any of you take Physics 30, I teach a whole couple sections on this in Physics 30. Here's basically like a very, very brief concept here. There's a lens right here, there's a lens right here. You guys see these 2 different lenses right here? And what ends up happening is this object right here is what it normally would be, and we're able to make it look like this smaller object right here by bending light through different lenses so that you can see something different. That's kind of the basic concept. And what you see is known as a virtual image, because you're seeing the shadows on the wall of Plato's caves. Are you actually seeing the cell? No, you're seeing an image of the cell. Okay. That's magnification. Most kids don't struggle with that. By the way, before I move on to resolution, I want to talk about contrast for a second. Most cells are colorless. Okay. Most cells have no color to them. And here's one of the biggest reasons why. We need to rediscuss from a previous unit, why do we see colors? When I'm wearing black, you're wearing red and purple and green. Why do you see those colors? Go ahead. Exactly, right? All light, white light, so red, orange, yellow, green, blue, purple, some of it gets absorbed by the red hoodie, except for red light reflects off of it. Well, when we're talking about cells, cells are so small that really they don't really absorb colors in that sort of way, right? Really, all light either goes through it or all light doesn't go through it. Usually it reflects all light colors, right? Because what does white mean? A white cell means that all light is reflected. Black means that all color is absorbed. And so most of these cells look like this, actually. This is a good picture of what cells normally look like. They're colorless. Light just kind of passes right through them. They're transparent. Okay? So we often use what's known as contrast, or a better word, we call it a stain. Okay? And there are two chemicals. I was even telling you about this earlier. There's two chemicals we use. One is called iodide. That's what this one does. Iodide basically makes them bleed into a purple color. And methylene blue is what we're going to use. It makes them look blue. Okay. But here's the most important part. It's only possible with cells which are dead. They have to be dead cells. You can't use living tissues. And the reason why is the cell membrane. So, quick aside. From grade 7, 8, whenever you learn about cells, does anyone remember what a cell membrane does? What's the point of a cell membrane? Okay, I'll share with you. A cell membrane is basically like the protective wrap that goes around the outside of the cell. It's the barrier that lets things in. The best analogy I would give for it, it's the gatekeeper. It allows things into the cell that need to go into the cell, like oxygen and sugars. It also sends things out of the cells, like carbon dioxide. Okay? And so it's the gatekeeper. It's basically, it's the guards. Okay? Well, if you have a living cell, the cell membrane is still functioning. And so therefore, when you add these dyes right here, the dyes can't go inside the cell because the force field is up. Is that a good analogy? The force field is up, therefore nothing can make it inside the cell. But kill the cell, have the cell be dead, well, now the mitochondria is no longer powering the cell's no longer got a nucleus that's functioning, cell is dead. Well, now the force field's down, now the dye can go inside the cell, and it makes it easier to see sometimes. Now, it makes the whole cell purple, or in this particular case, it makes the whole cell blue, but it just makes it a little bit easier to see. This is what you're probably going to see on Friday when we do onion cells. This is a really classic high school experiment. And what's cool about onion cells is you can really see two structures quite well. One is right here, you can see the cell walls. That's what you're seeing right here. These edges right here are cell walls made of cellulose. And right here, this thing right here is the nucleus. And so at the very least, you can actually see two organelles, and it's a lot easier to see because of the dye. If it wasn't for the dye, it would just be a little bit harder for you to be able to notice. Okay. So that's one of the techniques we use, is we use a contrast, a stain, or we use the word dye. But it's only useful if the cell is dead. If the cell is alive, the dye can't go in the cell. It won't let it. Okay. Now, here's the main slide I want to discuss, though. Why can't we just keep zooming in more and more and more? And it has to do with resolution. The best example I can give you is the idea of pixels. Okay? Microscopes are limited based on the same sort of concept of pixels. And it's really about this ability to distinguish between structures that are close together. The human eye, without any help, can distinguish basically a grain of salt, a hair on your head. But anything smaller than that, you can't tell the difference between the two. It just kind of all blurs together. And that's like your pixel rate. It wasn't until the 1870s that Ernest Abbe discovered that the resolution of a microscope is based on visible light, which makes sense, because why are we even seeing these cells in the microscope? Well, because visible light is what lets us see them. Can you guys see X-rays? Can you see infrared? Can you see radio waves? Well, no. Here's kind of a picture of something that we've talked about earlier in the course. Here's infrared, here's ultraviolet, and here's visible light. And the only thing that we can see with our eyes, because our eyes is what limits things, we can see from 400 to 700 nanometers. That's all that we are physically capable of seeing. I cannot see ultraviolet A. I cannot see infrared A. Does that make sense? So based on our receptors of our eyes, this is our limit. And Ernest Abbe did some math calculations and finally concluded this. Microscopes cannot produce images of objects that are smaller than light waves itself. If the object you're trying to look at is smaller than the wave, you won't be able to see it. And the wavelength of light, if you guys can't see this right here, goes from 400 to 700 nanometers. So basically that is as small as we're able to see. And he actually was able to prove that you can see basically half of that wavelength. That was what he proved. Half the wavelength of light is what the human eye has the ability to distinguish of. Anything smaller than that, and it doesn't matter how much you zoom it in, we can't see it. That's what I've got on this slide right here. Here's a picture of two things. I found this on the internet somewhere. This, I believe, is like leaves of something growing up, say, like out of a pond. And zoomed further back, I can kind of see individual strands. Like here's a leaf, and here's another leaf, and here's another little leaf. I can see individual pictures. The resolution on this picture is enough that I can individually make out this is different than that. Fair enough? This is the same picture, zoomed in 64 times, 64 times the zoom. And I can't see anything because it's, I mean, did I zoom in? Absolutely. But like, I really can't distinguish this particle right here from the one next to it. They all kind of look the same at that point there. And that's based on the wavelength of light. So here's the math behind it. Since wavelengths of light go from 400 to 700 nanometers, and Abbe discovered that we can only see things that are half that big because we see with light, right? Light microscopes can only distinguish to about 200 nanometers in size. Does that make sense? Because 200 is take 400 and cut it in half. And just for some practice in physics, what is 200 nanometers? An N, standing for nano, is times 10 to the minus 9. And if you want to write that out, it's this number right here, 0.000000200 meters. That is as small as a light microscope can be. You could add 1,000,000 lenses and try to zoom in that much. What limits our ability to see things? Our eyes. Our eyes see in colors. If our eyes could see ultraviolet, that would change things. If our eyes could see with X-rays, that would change things. Superman can use a better microscope because he can see with X-ray vision. Being silly, right? So, but we're limited based on what we're allowed to see because that's how we perceive things. By the way, I need to do a little brief aside here. There's actually a difference, though, between things being in and out of focus and having a lower resolution. And it may seem like it's really the same thing here, but you know how you can have an object that's kind of blurry? As a basic example, I'm going to stand up on the top of the screen here, and I'm going to fiddle with this dial right here. See how when I'm fiddling with this dial right here, I'm not changing the resolution. I'm just changing whether it's in focus or not. Okay. On the microscope, I kind of got away from talking about the microscope, but on the microscope, there are two different buttons or knobs. There's this one right here and this one right here, the coarse and fine adjustment knob. And what they do is they change whether the object is in focus or not. What these things do is they take the stage of the microscope and they move it up or down. That's all they do. So if you move this one right here, it moves the stage up or down, and that just basically changes the focus. But focus is not the same as resolution. They work the same to us because in both cases they're blurry. But if I can go back to this picture, here are some cells that I found in a picture online. And here they are out of focus. The lens is just blurry. All you have to do is just move that object either closer to or further away from the lens, and it'll bring it into focus. Resolution is more about this picture right here. This picture right here might have 1,000 dots per square inch, and this one right here might only have 100 dpi. You guys ever heard of that before, dpi? Printers and pictures will often have a certain amount of resolution. It's how many individual little pixels you have. And the better the picture that you have on your screen, if you have a 4K TV, it means you have more dots, and so you have better resolution. Okay. So here's kind of a summary of what I've just discussed then. The naked eye, so basically no telescopes, no microscopes, you can see 1/10 of a millimeter. Anything smaller than that, and you just see the object as a single shape. As an example, this white pile of salt right here, you can't see the difference in, unless you're like really, really close. Okay? But light microscopes have a practical limit, which meant that in the early part of the 20th century, biology kind of reached a standstill for a while based on microscopes because they couldn't zoom in any closer. However, as I've been sharing with you already, we actually do have the ability to have even better quality microscopes, and they're called the electron microscopes. So I'm going to spend a few minutes now talking about electron microscopes. Before I get to that, though, I need to let you know that this didn't happen until physicists discovered an important principle. It's called wave-particle duality. That deserves a lesson all on its own, and I do that with Physics 30 kids. But what physicists discovered is that electrons have the ability to be waves. You'll just have to trust me on that one for now. Electrons have the ability to act as waves. And because electrons can act as waves, it means that we're no longer limited to the 400 to 700 nanometers. We actually have the ability to go a little bit smaller on the spectrum based on electron microscopes. Okay? Just as a frame of reference, this is what electron microscopes have the ability of doing. This is a flea, I believe. And right here is the original picture of a flea. And then this electron microscope, hey guys, was able to zoom in on this little section right here that I put in yellow. And you can actually see the individual hairs on the flea. It is incredible the detail you get. Do you guys currently have a resolution that you can see individual hairs right here? I don't know. Like if I came really close up here, I might be able to say there's one there, one there, one there. I really can't tell, right? But this has such a better resolution. It's to the micrometers in terms of where it's at. It's incredible the zoom these things have. Okay, so there are two main types of electron microscopes. There's one that's called a transmission electron microscope, one that's called a scanning electron microscope. And on this slide, I actually have two different types of scanning electron microscopes. What basically happens is you shoot electrons at stuff. And I think the best way of me describing this is to actually fast forward in my slides and show you some pictures here. So I'm just going to find you this picture right here. Okay. This is known as a transmission electron microscope. Here's what you do. You take a focused beam of electrons and you fire electrons through a sample of stuff. Okay? And maybe the best example I would give you is it's kind of like a shadow. Imagine that I were standing against the wall right here and you were to shine a big light at me. If you were to look at the wall behind me, my shadow would make it so that behind me there would be a dark section where light didn't get through. That's basically what happens with these electrons right here. You shoot electrons at the sample, and if the electrons go through and hit this screen at the background, well, if the electrons didn't make it through, then you basically have the shadow. And if the electrons did make it through, then you get kind of the open section. You see how it's basically about whether objects make it through or not. One of the examples I gave is it's kind of like how a blind person would need to use kind of a stick in order to feel in front of them for things. If when you're feeling with your stick in front of you, you don't feel anything, well, then feel free to keep walking, right? Because there's not anything there for you to run into. And that's more or less what this does, is that the electrons might go through. A scanning electron microscope is kind of the opposite. When that person is feeling around, trying to see whether there's something in front of them for them to hit with their little walking stick, if they bump into something, that's the scanning electron microscope. Here the electrons do a very similar process. You focus beams of electrons very narrowly at a sample, but you bounce them. You don't want them to go through. You bounce the electrons off of the sample towards this detector. And as they bounce towards this detector, it gives you a picture of how these things ricocheted. Okay. It's binary code in a way. You either get something that ricochets or something that doesn't ricochet. It's either it transmits or it does not. It either passes through or it doesn't. This one's all about bouncing. Okay. So if I can go back a slide or two here to where these originally were, where was it here? Here's basically the concept. A transmission electron microscope passes electrons through a specimen. Makes sense, transmission means through. And you get two-dimensional images that are up to 100,000 times zoomed in. What was the limit of light microscopes? Remember what light microscopes were able to do? It was a 1250 to about 1500 times zoom. The transmission electron microscope now lets you go to 100,000 times more zoomed in. That's incredible. That's way more zoom. With the resolution, you need to be able to see things. Scanning electron microscopes. Now, I wish I knew more about these two things. I'm not a biologist. I'm a physicist. But there's two different types of scanning electron microscopes. I mean, basically what they do is they scan the surface of an object and they make a 3D image. And there's two different types. I don't know really why they're that much different from each other. But what I do know is the scanning tunneling microscope can get up to about 2 million times zoom. So much so that we can actually see DNA, which we're going to talk about more over the next couple of classes as to why that's so relevant. Us being able to actually look at whatever you consider looking at to be. Because again, these are the shadows on the wall, right? We're not actually going and looking at real DNA. We're bouncing electrons off of it and getting a picture. But the bounced off picture of DNA may look something like this. These are some of the pictures I've got right here of what electron microscopes can do. And it is incredible. This is a flea, I believe. Look at the detail on that, hey? The resolution, the detail on this is incredible. But like these are viruses. These are germs and bacteria that we're able to see. We can actually see the receptor locations on the edge of this so that we can try to figure out exactly how these viruses bind to cells. So that if we can remove the binding capabilities, we can keep people from getting sick. I mean, it's incredible the detail that we can see with these electron microscopes. And again, the point I want to make with these slides right here is that it's all about whether electrons go through or whether electrons bounce off. Okay? The electron machine that measures it is basically a computer that records in binary. You guys know what binary is, where it's like a 1 or a 0, it's on or off, right? This one right here I think is the best way of describing it. Either the electron goes through or it doesn't, right? And based on that, the binary code that ends up getting shown on the bottom of the screen here basically makes a computer signal that computers then use to make an image. There's not actually a picture that shows up here. This is why I like using Plato's cave analogy, because you're really just seeing shadows. This object right here that's in the way, the electrons are just phasing through it. You're just getting a picture of a computer code. Here, let's talk about these slides right here. This right here is one picture of an electron microscope. And this one right here is a second picture of an electron microscope. And the reason I put two of them in here is just to discuss the concept of resolution. I found two pictures on the internet. Which one of these pictures, here's one, and here's two, which of these pictures do you think has better resolution? One or two? I'd probably say two. Because like, look at this picture right here. I found this on the internet and I blew it up really big. And so this has been magnified. But can you guys see how it's pretty fuzzy? Like right here, take a look at this wire right here. Like, can you see how it's kind of fuzzy around the edges there? Right? There's a little bit of distortion up here. As you take a picture and blow it up bigger and bigger and bigger, the resolution may not match it. And if you blow a picture up bigger and bigger and bigger, you may not actually be able to tell the differences between different sides here. I think this particular picture is a little bit better in its resolution. That being said, look at this scientist's hair right here. Can you actually see individual strands of hair? Well, no. But I can tell you these markers on the wall, I can see the differences between them. Right? So anyways, I really want to make sure you understand that magnification is not resolution. Okay, let's go back here and talk about the differences between the two. If you'd like to, I'm now really talking about number 3, the advantages and disadvantages to like light versus electron microscopes. And the initial question you might be asking is, Chris, why don't we just use electron microscopes? They sound awesome. They sound like they're way better. Well, yeah, but there's some pros and cons. So here's a couple. Number 1, light microscopes can view images in color. You may have to add contrast to make it show up, but you can see things in color. But electron microscopes, you only get monochrome images. The reason why is that you're not really getting a picture. It's not like taking a camera picture. It's shooting electrons at stuff. And if the electrons go through or bounce off of it, they then get sent to a computer, which then using binary code basically says yes or no to a black or white pixel. And so that's why those pictures are monochromatic. They're never, ever going to possibly be in color, because that's not how it works. There's shadows on a wall, right? Okay. Here's another problem. Light microscopes, you can actually see a large field of view, whereas electron microscopes are a limited field of view. You can't shoot electrons at a large object. It's just not reasonable, right? Like, I cannot use a microscope to take an electron image of a human person. That's just, there's way too much of you to measure. Does that make sense? So it has a very limited, narrow scope. In terms of the picture right here, here's a picture of an electron microscope. It has a high voltage electron gun, and it has condenser lenses. These condensers are basically narrowing the field of electrons to like the tiniest points possible. You can't have that be super wide. It actually has to be exceptionally narrow. So we call that a field of view. And then here's the other pro to a light microscope. They're cheap and easy, whereas electron microscopes are difficult and expensive. That being said, there are some advantages to the electron microscopes. Oh, hang on, I got one more the other way around. Light microscopes, by the way, you can view living objects though. But electron microscopes, they need to be dead, because you're firing electrons at them. And you won't learn this again unless you take some physics with me. But electrons being fired in a gun, we actually have another name for that. We call that beta radiation. We call those beta particles. And you're basically shooting radioactive radiation through this material. So it's not going to be alive. If it is alive, you're going to kill it. So there is a disadvantage to electron microscopes. No color, very narrow view, very expensive, dead objects only. Those are the downsides. Plus sides, huge magnification. 500,000 to 2 million magnification. The resolution is tiny. Okay, you need to know these prefixes here. The resolution of a light microscope is in the micros. The resolution of the electron microscope is in the nanos. And nanos are over 1,000 times smaller in terms of what you can see. The comparison is basically this. Take a rock and compare it to Mount Everest. Well, that's what an electron microscope can do compared to regular light. Like, the scope of what electron microscopes can do is insane compared to light microscopes. And it has led to some incredible advantages and advents and increases in microscope technology that have really affected what biologists study. Here's another picture here that just kind of describes what we're able to see. I like this slide. I found this online. I stole it. The human eye. What are you able to see? We can see people. We can see a hand. We can see a finger. And you might just be able to see human hair. That's basically the limit of what the human eye can distinguish. Okay, I can see your hair, but I can't see individual strands anymore unless I'm like really close, right? Well, with a light microscope, you could see human hair. You could even see a red blood cell. You could see a bacteria. However, it wasn't until electron microscopes came around that these things could be studied, specifically viruses and DNA. And we're going to finish off my last couple of slides discussing why that was so important, because being able to see viruses allows biologists the ability to give us drugs, the good kind of drugs, right? The ones that are going to suppress disease, the ones that are going to suppress like antibiotics, right? Those sorts of drugs. I have to use that word very carefully, right? DNA, the ability to see DNA allows us to recognize that if there's a person who, say, has a problem genetically with their mitochondria, now we can actually do something about it. And if it wasn't for electron microscopes, we would never have reached these limits down here to be able to see even atoms themselves. It's actually very challenging to see atoms themselves, though, because how does the electron microscope work? It shoots electrons. Well, if atoms are made of electrons, you actually can't even get a picture of an electron with an electron gun, because that really just doesn't really work. Sorry, my microphone's falling off here. Right? Let's say that you wanted to shoot electrons at atoms themselves. That's basically our limit right now. If you shoot an electron at an atom, you can't actually see another electron by shooting an electron at it, because as soon as the electron hits the other one, it moves it. So there's some practicality problems there. Okay, I think I've talked about all these slides here. Okay, here's our last discussion topic that I've got for you for notes to write on here. We want to talk about some other techniques that scientists have for being able to get a good, clear image with microscopes. Okay. I've already discussed one with you. It's the technique called staining. If you add a stain or a color or a dye, you can see better your clear, colorless cells. Okay. Here's two more. This one's known as a centrifuge. What a centrifuge does is it spins around in circles. And you might have seen a machine like this before on like a crime TV drama like CSI or NCIS, you know, where they're like doing DNA samples. They put a sample in a little test tube and they spin it around in a circle. I mean, I might say, why, Chris? What does that accomplish? Well, it's all about density and size. If you have a test tube that you spin around in a circle, it's going to separate it out into layers where the heaviest layer will be at the bottom. And let me just do some new colors here. One of the few times I'm going to draw this unit here. And then maybe the medium cells or whatever it is you have will go in that layer there. And then the lightest and least dense cells will probably float at the top. And it's a good way to separate out materials. Yeah. That's exactly where I was going to go next. We've actually discussed this concept. They're known as suspensions. You guys remember talking about suspensions in chemistry, not the you get suspended from school suspension, but like the salad dressing, the oil and water, right? You separate it out into layers, and here's why that's useful. Let's say you're taking a sample of the human body and you want to look at nerve cells. If you were to just take a sample with a scalpel and just scrape some off, you're not going to just get nerve cells. You're going to get muscle cells. You're going to get skin cells. You're going to get heart cells. You're going to get all sorts of different cells. You're not going to get just one kind. Does that make sense? So we have to try to find a way to separate it out so that you get just one to look at. Otherwise, it's really hard to differentiate between the two. So the centrifuge spins it around in circles, makes it so that they separate based on density, and then you can use a pipette and extract only the one type of cell you want. Really good idea. Second thing that I'd like you to know about. I wish I knew more, because I'm not actually a biologist. I didn't go to school for biology. I went to school for physics and chemistry. But this one's pretty cool. It's called a green fluorescent protein, acronym GFP. And it's found in luminescent jellyfish. You guys have probably seen these before in like movies. My daughters watched the one called Sing. You guys ever watched that one movie where like the koala bear makes all the jellyfish light up the stage underneath it? Anyways, if you haven't seen the movie, don't worry about it. But like jellyfish have the ability to like glow in the dark basically, which is super awesome. And the reason why is that they have a protein in the jellyfish that is luminescent. That's the word we use. The protein glows green when you expose it to ultraviolet light. Here's what's awesome about this. You don't have to kill the cell to see it now. You attach this jellyfish protein to your cell, you let the cells grow and multiply, and then when you want to look at the cells under a microscope, they will glow green, just like the jellyfish's cells will, but they're still alive. So now you don't need to kill them to see them. Because what do you have to do with the stain? The stain needs the cell membrane to be dead, otherwise the stain won't go inside. These proteins can be attached to the surface of the cells, and it doesn't actually have to kill them. So it allows for contrast in living cells. Really cool advent, based on basically taking jellyfish DNA and adding it to the cells you're looking at. Sweet, eh? All right, last couple of slides. So all of this is really to get to this point right here, the question of why should we care? I mean, it's cool to look at stuff under microscopes, I guess. I mean, it's fascinating. But here are 5 really important reasons and things that have happened because of the ability to look through microscopes with the degree of electron microscopes that have really affected things. Here's number 1. It has allowed us to begin what's known as the Human Genome Project. Maybe you've heard of this before. What they're really trying to do is code the entire human genome, all of the DNA in a human body, through what we call gene mapping. Fellas in the back, please. Maybe you've heard of these 4 bases before. They're called adenine, cytosine, guanine, and thymine. You maybe learned about them in grade 9, maybe not, I don't know. If not, don't worry about it. But we abbreviate them with these letters here, A, T, C, and G. And all of your DNA, all of your genes can basically be coded into replicating patterns of A's, T's, C's, A's, G's in these unique patterns. It's almost like binary. You know how binary is zeros and ones? The human body, if we really wanted to break it down, is really 4 binary, whatever that would be, quaternary. There's 1s, 2s, 3s, and 4s, A's, T's, C's, and G's. And scientists are really trying to figure out whether they can actually find the entire code of the human body, which is amazing because they can actually see it using these electron microscopes. Now, why would we want to do that? I mean, yes, there's discussions on things like cloning and being able to do those sorts of things, but here are 4 main reasons. Number 1, we know now about degenerative diseases. 3 of the biggest ones, these aren't the only ones, but 3 big ones, are Huntington's, Alzheimer's, and Parkinson's. Okay. If you know anything about these diseases, probably the 2 things you know are 1, they're genetic. And that like, I know that I'm not probably going to have Parkinson's because I don't have a family member who also has Parkinson's, right? It's passed down through DNA, through moms or dads, right? But 2, what we recognize is that for whatever reason, these genetic diseases cause nerve and brain cells to break down. It causes memory loss. It causes people to have muscles that just don't control the way that you want them to. And so we recognize that there are problems, there are whatever you want to call that, but there's an issue in DNA that from one generation to the next is getting passed down incorrectly. And just like I mentioned with the idea of inserting a 3rd DNA into a baby, changing out a mom's mitochondrial DNA, if scientists can figure this out, maybe one day we can find a cure for diseases like Parkinson's, Huntington's, Alzheimer's. Similarly, we may be able to find a cure for cancer through this sort of process, slowly but surely. If you're not really sure what cancer is, I mean, you probably heard the term before, but what cancer is cancer is cells that grow uncontrollably. Okay? They end up forming tumors. Cells are supposed to grow. You are supposed to replicate. Okay? There's 2 types of ways that humans can basically replicate. One is sexually, one is asexually. Okay, bit of review from grade 9. Sexual reproduction is where, it's typically a mommy and a daddy love each other very much. Like, okay, sperm and egg, that's one way that you can have reproduction. But all of you right now are asexually reproducing in the way that your bodies are growing. You're having new hair cells, new blood cells, new bone marrow cells are being grown every minute, right? The human body replaces itself, I think, every 7 years or something. Like the cells that you have in your body are not the same cells that you're always going to have. Okay? Long story short, as your cells grow, cancer cells grow without restrictions, and they just keep growing and growing and growing, making masses and lumps, which we call tumors. If scientists can find a way to figure out why they're doing this and find a way to solve that problem on a molecular level, hopefully one day we'll find cures for cancer. Okay. Here's another big advent of electron microscopes. It's the field of pathology. It's the study of disease. Viruses, things that make us sick. Right now is a great example. It's cold and flu season. When we give people their flu shots, we're trying to find a way to make it so that the viruses that we believe are going to be around during this flu season, we find a way to prevent them from basically connecting to cells. Okay? I showed you these pictures way back here. Let's find them again here. Of viruses and bacteria. See all these little like bumps on the edge of these? Those are receptors. And different bacteria have the ability to like attach to these little attachment sites. If we can find a way to make it so that the attachments don't happen, we can hopefully prevent the spread of disease. And so pathology is basically the study of viruses, which, by the way, I want to point this out. Viruses are not considered cells because they're not alive. Now you might say, but they replicate. They do. But they need a host. A virus needs to infect a host, and then it can replicate. So on its own, a virus is not usually considered to be alive. It's actually not considered to be a cell, although it basically is a cell. A virus basically is everything like a cell, except it has to invade a true cell and infect it. Okay. Final topic, then I'm finally done. I'll be quiet for the day here. A lot of me talking here. Stem cells may be the key to all of this. Okay. If you know much about stem cells, they are basically blank cells. They have not specialized yet. The analogy I gave the last class is, say you go off to university and you don't know what you want to be yet. And so when they ask you what your major is, you say you are undeclared or you're in general studies. You ever heard of maybe that phrasing before? But maybe one day you decide I'm going to be a math major, a music major, an archaeology major, a management major. Now you're specialized. When a human is first conceived as an embryo, when sperm meets egg, the cells that are formed are stem cells. They don't actually have a specific function yet. They aren't heart cells. They aren't brain cells. They aren't nerve cells. And so pardon me, stem cells are blank. Eventually they'll become muscle or fat or bone or blood or sex cells, whatever they become. Yeah.
Last year when we talked about stem cells, we actually talked about how scientists are trying to use them to help regenerate.
Exactly. And that's actually kind of, these things all kind of fit together, is that these may be the key to solving those, right? And this is where we're going to do that case study on Thursday. There's then the ethics of it is, to what degree should we be tinkering with our DNA? It's one thing for me to go to the doctor and say, doctor, I broke my leg, put it in a cast. It's another thing, I think, for me to go to the doctor and say, doctor, I have a cough. Can I have some medication to suppress the cough? I'm not saying we shouldn't. Please don't go home and say, Chris says that we should let all the cancer patients. I'm not saying that. But to what degree should we be monkeying around with DNA to try to solve other problems? Like that's a deeper concern. Like what happens generations in the future when we alter our DNA? I'm not saying that we shouldn't definitely look into it, but that is a huge ethical, societal challenge. So that's why we're going to do that case study on Thursday. Okay, thank you guys for being patient with me for my lecture. Let me pause this and let me give you some final remarks before we leave here. So that's my lesson.